Now, one of the most recent fields of research in stem cells has been in tissue-specific stem cells. It's very interesting to find that almost every organ in your body has stem cells in it. It's not just your bone marrow. For instance, the heart has stem cells. Unfortunately, they're very rare. Only 1 in every 40,000 heart cells is a stem cell. However, some researchers at Cedars now have been able to biopsy the heart, grow the cardiac stem cells, and now are actually doing clinical trials on treating heart attack patients with these cardiac-derived stem cells. The brain has stem cells. They're rare. They're found in the subventricular zone, and I'll show you where that is here in a second. But these cells are not pluripotent in the sense that the other cells that we talked about. So they don't necessarily have a CD34 plus staining. But what the researchers have found, these cells still can repair, regenerate, and heal nervous tissue. And for that reason, other markers besides CD34 plus have been used for these tissue-specific stem cells. The liver also has stem cells. These were not discovered till just a few years ago, and they're very hard to find. Here's a picture of fluorescence antibody tagging of a cell marker on what's called oval cells of the liver. The kidney also has stem cells. The problem with the kidney stem cells is there aren't any good markers right now for these stem cells. The pancreas has stem cells in it. And here's a picture of a fluorescent antibody labeled pancreas cell. The intestines have stem cells in them. These are found in the bases of the crypts. The lung even has stem cells. And the interesting thing is that we now believe that it may be these stem cells in these organs that may form cancer. So not only are stem cells a very exciting area for treating disease, treating aging, but it may unlock some of the secrets for drug resistance and for recurrent cancer. Now, with that in mind, I want to just give you a few examples of these stem cells from specific organs. For instance, cardiac stem cells. Right now, the hope is that these will be a treatment for a heart attack. Here's a diagram that shows you the progenitor cells that can be harvested from the atrium or the ventricle, the ones below. And these cells have the capability of self-renewal, like stem cells do. And they can then be differentiated and cultured in the lab into cardiac stem cells. I had the chance to attend a lecture that Dr. Marban gave on these. He's one of the researchers at Cedars. And I'll tell you the thing that just amazed me more than anything I've ever seen. If you take these stem cells in a petri dish and you have a low concentration of these cells, they just sit there and they look like any other cells. As the concentration of the cells gets higher, they start to clump. And when the concentration gets high enough, if you stimulate that petri dish with a very low and a proper electrical current, guess what happens? They spontaneously start contracting. And when you see these cells just spontaneously beating there, it is the most amazing thing I've ever seen. And it raises the question of why are they beating? I think it's because they were programmed to be cardiac stem cells. And that's the whole point behind tissue-specific stem cells. We think that they are already partway down the programming pathway, and that's why they are so exciting. Now, with that in mind, there are a lot of problems, too. The biggest problem for cardiac is these cells are rare, and because of that, if you have a heart attack, you're not going to have enough stem cells in the zone of ischemia or the zone of necrosis to repair that muscle. So how do we get enough stem cells into that? And this then shows you how difficult using stem cells in. This next slide shows you all the steps that the researchers are going through trying to take a stem cell and actually making it into a cell that contracts in the damaged heart. The pictures at the top are the methods, and we haven't even decided for sure what is the best method. And the words at the bottom are the challenges. So I'm going to just briefly go through this. The first step is stem cell isolation and culture. Here, we're not sure what the best method is. Is the best method isolating stem cells from the bone? Should we take them from the blood? What about a muscle biopsy, a skeletal muscle biopsy? At UCLA, I had the chance to work with several of the cardiologists in the 1990s where they were growing skeletal muscle cells and then re-injecting those into damaged myocardium and trying to see if that would improve the ejection fractions of the heart after this therapy. My job in the research was to biopsy the muscle. We then sent these cells off to a laboratory in North Carolina. There they would grow these skeletal muscle cells, would send them back to UCLA, and the cardiac surgeons would inject them into the damaged myocardium during coronary bypass surgery. And the initial results were quite promising. They did show improvements in contractility, but in the long run it didn't make any difference. So step one alone is a very difficult thing, and that's why researchers now believe that probably cardiac stem cells may be the best treatment for cardiac disease. The next hard thing to decide on is stem cell delivery. What's the best way to give these? Should we be giving these cells intravenously? Should we be injecting them into the coronary arteries, or should we be injecting them directly into the muscle? The next step is stem cell survival. Most of the studies have shown if you inject, let's say, 100 million stem cells, possibly only 10% of those cells actually survive. The large majority of them may never actually make it. Even those that survive, the question is, are you going to get those cells to contract synchronously with the other cells? And I think this helps you realize what the challenges are with trying to get stem cells to turn into a functional heart muscle. Basically, it ain't easy. That's all you need to remember from this slide. Now, brain cells have stem cells, and there's been a lot of hope that this may end up being a treatment for Alzheimer's, spinal cord injury, stroke, degenerative diseases, multiple sclerosis. And what we now know is that the brain itself has stem cells. They're located in the periventricular area around the ventricles of the brain. And again, the problem here is that with most of these diseases, there's a lot of damage. And how do you get these stem cells to recreate all that damaged tissue? So in short, if you think that making a heart is difficult, probably making a brain is 100 times more difficult than making new heart muscle. The liver also has stem cells. We think that most of these stem cells are located around the blood vessels in the liver. These cells are very easy to find in the fetal or infant liver, but they're very hard to find in adult. In fact, they weren't found until just a few years ago. They now believe that the probable cell that is a hepatic stem cell is a oval cell or ovalocyte. That's different than the ovalocytes you see on a peripheral blood smear. These stem cells can form two types of cells, hepatocytes and biliary cells. And these cells also have the capability for renewal. Again, these cells don't have the classic CD34 plus marker, but researchers have found 3 other markers that can be used to identify these cells. Again, the problem here is just because you have these stem cells doesn't mean you're going to have a new liver. Damaged liver is very scarred, and for this reason, adding stem cells is not going to undo the scarring that's happened from years of alcohol use or viral infections or other things like that.